Hello, everyone. Welcome to the BPI Spring Digital Week. My name is Eden Turner, and I am one of the senior content producers with Informa for our Bioprocess International series. And I will be your host for today's session titled Transforming Upstream Bioprocessing, Rapidly Optimize Viral Vector Production Without Process Disruption. Before we kick off, though, I have got a few housekeeping items to go through with you. So if you do experience any difficulties with audio or advancing of the slides, refresh your screen with F5. If you're experiencing any other issues, please hit the question mark button to receive assistance. At any time during the presentation, please submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. You can also download a few featured articles in the resource list box on the right-hand side of your screen. Now, I would like to introduce our speaker for today. We have John David de Jong, Vice President of Scientific Operations at Virica Biotech. Thank you so much for joining us today, John David. I would now like to hand it over to you to begin your presentation.
Perfect. Thank you for that very kind introduction, Eden, and welcome everybody. Today we're going to be talking a little bit about transforming upstream bioprocessing and really with a focus on viral vector production. And this will be centered around what Virica brings to the table, which is our unique VSE technology, which I will introduce in a second. So just kind of going through quickly the webinar agenda, we'll talk about some of the challenges that folks face in the cell and gene therapy space, especially around biomanufacturing. I'll introduce you to the concept of viral sensitizers, or VSE technology. This is the core technology we have here at Virica. And then we're going to go through a few case studies about curated VSEs for the gene therapy market, primarily focused on AAV, curated VSEs for the cell therapy market. And this one gets a little bit interesting because we will talk about not only lentiviral production and enhancing that, but also the concept of using the VSE technology to sensitize the end product, so the actual cell to transduction. So we will touch on that a little bit. And then finally, we'll talk about some of the services and cost modeling that we do here at Virica, and then the ways of working together. And then obviously, finally, we will end off with some questions and answers. So really just quickly about Virica Biotech, we really do view ourselves as cell enhancing experts. And what we mean by that is using our core technology to really set up the cell from a biological angle for success when it comes to biomanufacturing these next generation therapeutics. And so this really does empower the advancement of cell and gene therapies and vaccines through acceleration of the manufacturing process, but also increasing productivity and reducing that cost, something that's obviously sensitive, especially within the cell and gene therapy space. What sets us apart a little bit is the unique angle we take here, and that's by leveraging our proprietary set of viral sensitizers, VSEs, which are actually small molecules which will transiently attenuate biological processes within the cells to, again, increase biomanufacturing. And then a byproduct of this is we've developed a lot of expertise now in what we're terming high throughput virology workflows and design of experiment methodologies. And we'll touch on those a little bit later in the webinar. So obviously, everyone wants to have a very efficient process. And this is especially true when it comes to AAV, lentivirus, and cell therapy manufacturing, but is equally true for replicating modalities such as vaccines. And there's lots of different ways to approach process intensification. A lot of them from an engineering perspective through bioreactor design. There's also cell and vector design to get more efficient productivity, and bespoke medias and media optimizations. Where Virica comes at it is from some of those, as I alluded to earlier, fundamental biological barriers, which might be acting as bottlenecks to really efficient yields when it comes to the quantity as well as the quality of the vector that comes out the other end of these manufacturing modalities. One of these areas, most definitely, is the innate cellular defenses, which are present within these cell lines. And it's not something that we always think about, but essentially, we're all alive today on this call because we have an immune system. And most people are very familiar with the adaptive immune system. It underpins modern vaccination strategies, antibodies, T cells, all that other good stuff. But there's a whole other arm to the immune system, and that is the innate immune system. And this is kind of the one that's working in the background. It's mopping up 99.99% of what we encounter throughout our daily lives. And one of the components of that innate immune system is that every cell has the intrinsic ability to sense pathogens, respond to them, and mount a response that's there to blunt the replication of those pathogens. And so what we have here on this slide is just a cartoon diagram of a cell. And it's important to understand the concept of the PAMPs, the pathogen-associated molecular patterns. These are molecular patterns that our cells have evolved to detect and respond to. And usually this comes in the source of the introduction of viruses, for example. That incoming virus, usually triggered through nucleic acid sensors, triggers a whole set of pathways and effector molecules within the cell that are there to blunt that viral infection. The same holds true for reverse genetic systems. So that incoming plasmid DNA or incoming nucleic acid, however you're delivering it to the cell, is still sensed by the cell and recognized as a pathogen, and the cell will mount a response. So really what happens is this activates key signaling pathways, and we've highlighted a few of them in those kind of green ovals there, which then activate what we are calling the innate cellular defenses. So essentially activation, sensing of that nucleic acid, causes a signal transduction cascade through those signaling pathways. And it culminates in the expression of a multitude of effector molecules, which are there to blunt viral infection and production. And this really, what this means is reduced transfection or transduction efficiencies depending on the applications. And we're going to talk about a few of those applications in a little bit. So really, in a nutshell, what we're talking about is some small molecules which could really have a beneficial and quite large impact on a biomanufacturing scenario. So here at Virica, we actually have a whole panel of VSEs, libraries of these, which can transiently attenuate some of those pathways I alluded to earlier. And since these pathways are very conserved across different cell types, and they are triggered by different viral families, we can draw on this library to hit different manufacturing modalities to try to increase productivity and yield in a very simple way. And so that's really through the integration of the VSEs in the upstream process. And it is very much, the VSEs are very much viewed as ancillary material to the manufacturing process. So you can think of it as a raw material. It's essentially a media additive that is generally added at the time of transfection or infection, depending on, again, how you're producing your viral vector. And we have a lot of evidence that suggests that these VSEs are actually quite quickly metabolized by the cells. And in fact, I'm going to take you through a few case studies where we demonstrate that they fall below detection levels of our sensitive analytical assays in the crude lysase, let alone anything you're doing to purify viral vectors. And the beauty of the modalities used to purify, or the strategies used to purify viral vectors, they're really built to keep the larger molecules and get rid of smaller molecules. So any residuals that may be present are easily removed with those downstream processes. So really, in a nutshell, adding the VSEs to your upstream process, you do not need to change anything within your platform. They seamlessly integrate and are considered ancillary raw materials. And of course, we never bring anything forward that we don't feel comfortable about. So that being said, we have DMFs filed for key molecules. If you're sitting in Europe, we have regulatory support files very much laid out like these DMFs. And importantly, we have those analytical assays ready for residual detection to support any GMP use of the VSEs. So that's a lot already, but I figured we'll take you through a little bit of a scientific journey to give you an idea of how the VSE technology was kind of thought up in the first place and discovered, because I think this is important to contextualize some of the further conversations we're going to have. So the VSEs actually come out of our co-founder and actually CEO's lab, Dr. Jean-Simon Diallo at the Ottawa Hospital Research Institute. And he was working on oncolytic viruses. So for those of you who don't know, oncolytic viruses are replicating viruses which preferentially replicate within the tumor and maybe not so much in normal cells. And the whole idea there is to debulk the tumor, but also draw the immune system's attention to that tumor to try to get an immune response or reawaken an immune response. What the folks in the oncolytic virus field had noted is that while some tumors responded very beautifully to oncolytic viruses, there were other tumors that were completely resistant to viral infection. And so what JS, the way he approached this is he basically wondered, are there small molecules out there which could sensitize the tumor to that viral infection or that oncolytic virus? So this is where we get those viral sensitizers, VSEs. And so he basically developed a high throughput screen based on, and this is actually the real world data shown on the left of that high throughput screen with an oncolytic virus expressing M. cherry. As you can see in the top left-hand corner, that's the control. So you can see these tumor cells are quite resistant to that viral infection, very little M. cherry expression. But pre-treating or treating at the time of infection, the cells with certain molecules very much sensitized them to that viral infection. And this is kind of where the VSE technology was born and came from. But it's equally applicable from that therapeutic angle as it is to the viral manufacturing process. And in fact, we've demonstrated that that original set of VSEs are quite good at increasing the yield, especially of replicating viruses. So what I'm going to take you through here very quickly is a collaboration we had with the Max Planck Institute with Yvonne Gensel et al. And here they were looking at a yellow fever-based backbone for vaccine production. And essentially, they were looking at 3 different cell lines. And if I draw your attention to the right-hand side there, in the green, there's the low producer cell line. In the light blue, the middle producer cell line, and then a high producing cell line in the dark blue. And they assessed the VSE molecules in a DoE type approach. And here you can see that baseline compared to the plus VSEs for each of those different cell lines, we were able to hit on a formulation of 1 or 2 molecules that gave anywhere between, you can see just above a 3-fold increase in that high producer cell line to almost a 7-fold or just above a 7-fold increase for that low producer cell line. So very much demonstrating the utility of these VSE molecules within the context of replicating viruses. But equal to that, we wanted to ask the question of, are they equally applicable in a reverse genetics type situation? So these would be the general modalities that are used for AAV and lentivirus manufacturing. And recently, a very nice paper came out from Eric Barton's group at Pfizer, where they took an omics approach to really map the response of suspension HEK cells in a 50-liter bioreactor to a triple transfection-based approach for AAV manufacturing. And in this particular study, one of the key sets of pathways, there are several pathways that are meaningfully upregulated, but one of the key pathways is the inflammatory and the interferon response. So as you can see on the right-hand side here, the incoming plasmid, they'd evoked a cellular response within that innate antiviral within the first few hours of that transfection process. And then this was followed by a wave of those effector molecules that I alluded to earlier. So an interferon response up front that unleashed those inflammatory and antiviral responses during the time or during the kinetics of the packaging and replication of the AAV vector. So this really does demonstrate that those key pathways that the VSEs were targeting are very much present, A, within suspension HEK lineages, and B, within the context of that reverse genetics AAV production. So with another collaboration partner, we looked at that initial set of VSEs that were curated on those oncolytic viruses for their ability to increase the production of AAV within suspension HEK cell lines. So we screened with this partner a panel of 8 VSEs. As you can see on the right-hand side, there were 3 key molecules that gave us what we were considering and what they were considering a meaningful benefit, which is above that 2x threshold. What we did with this partner is take the best performing molecule, which is VSE-A in this particular context, forward to both an amber system and a 5-liter bioreactor, So what's really important here for this AAV8 that we're demonstrating here, this is the bona fide therapeutic. So this is not a GFP reporter or a luciferase reporter. This is the bona fide therapeutic vector that in the AMBER system, we're able to achieve about a 2.2-fold enhancement in the genomic titer. When this was evaluated at the 5-liter scale, we were actually able to achieve a 3.8-fold increase in the genomic titer. And in this particular context, our increase in genomic titer outpaced our increase in physical ELISA. So what that actually resulted in is an additional 2.2 increase in the percent full. This has since been demonstrated at the 50-liter scale, and we're now moving on with this partner to the 250-liter scale to help support GMP manufacturing. So really there showing that that initial set of molecules was beneficial within certain contexts in AAV production. Additionally, with this particular, again, because this is moving towards the GMP, what we were able to show using our residual analytics is clearance of the molecules even in the crude harvest, as well as any of the downstream samples. So what I show you on the right-hand side on panel A, this is our analytics where we purposely spiked in the VSE, which we were able to pick up, as you can see that arrow demonstrating. And then in panel B, this is the crude harvest from a VSE treated bioreactor. And then you can see we cannot detect the VSE anymore. We fall below the detection level of this relatively sensitive HPLC-based assay. We also have more sensitive mass spec-based assays for these VSEs. And again, we demonstrated the same sort of clearance within the process. Now, something that we had noted as we started using those original set of VSEs that were, again, curated for oncolytic virus production, but did translate nicely to those reverse genetic systems, is that not every HEK lineage was responding in a positive manner to the VSEs. And so what I mean by that is that the HEK cells that I have growing in my lab are going to be very different than the HEK cells that are growing in someone else's lab, let alone any of these single cell clones that have been picked out, for example, your VPC 2.0s versus 293Fs versus proprietary cell line that a CDMO or therapeutic manufacturer might be developing. And what we had noted is when the VSEs did work, they worked very consistently, very nicely, and at scale. But with some of the lineages, there was essentially no benefit to the VSEs. So that prompted us to speculate that all these different HEK lineages, while they share a name, they do not necessarily wired the same. I.e., they have different pathways that might be active, and they respond differently to incoming nucleic acid in the VSEs. So what we did is we decided to expand our portfolio of VSEs available, especially within the context of AAV. And by the way, I'll preemptively note, we noted the same thing with lentivirus, this lineage-specific responsiveness. So what we wanted to do is bring more tools to the toolbox to help mitigate against that kind of scenario. So as I mentioned earlier, the initial set of VSEs were really curated on tumor cells with replicating viruses. So what we did is we backed up and we developed assays and it developed in particular a 96-well-based assay to screen specifically in the context of AAV in HEK cell lines. And so here what we did is we went ahead and we did a screen of over 16,000 molecules. What I'm showing you kind of on the left-hand side there is just a snapshot of some of that screen. And obviously, if we look at this graph, you can essentially treat the y-axis as statistical significance, and then the x-axis is fold change over baseline. The VSEs we're interested in, or the molecules we're interested in, are anything that shows up on that upper right quadrant, where we see over a 2-fold change that is statistically significant across the repeats that we did during the screen. And out of this, we've pulled out a set of VSEs, which we demonstrated utility at, not just at the small scale, but also at the bioreactor scale. So things that worked very well in our 96-well assay translated well, at least to the 250 mil bioreactors. And this is something we did with our friends at Culture Biosciences. Where using these molecules, VSE 7 and 8, we got anywhere between 2 and almost 4x enhancement in the VPC 2.0 lineage at the bioreactor scale with PEI-based reagents. And so leveraging this particular set of cells has allowed us to hit on formulations that can be developed for common commercial cell lines, but also proprietary cell lines. So what we're showing here is a collaboration we did with Virica in Italy, looking at their in-house proprietary suspension HEK cell line. So what you can see here on the right-hand side, top, VSE-A, which across two different serotypes gave that over twofold enhancement that we like to see with the VSEs. And then VSE-B, which again tested in only a single serotype in this particular case at Shake Flask, gave a just over twofold increase as well. What gets interesting here is when we get multiple hits like this, VSE-A and VSE-B, we can bring them together in that multi-compound formulation. And so what you can see here, again, on the left-hand side is this VSE-A plus B combo, where alone A gave about a 2.1 increase and B gave a 2.8-fold increase. But in combination, we were able to stack those increases together to achieve an over 4-fold increase in this particular cell line. So again, speaking to the value of being able to draw on the different tools we have, or this now new curated library specifically for AAV, and be able to stack the benefits of the molecules together. So really, this is what we're starting to lean into and call the Virica advantage. And this is curated sets of molecules for contextualized for very specific production scenarios. So I'm going to talk a little bit about that for AAV, then we'll talk about lentivirus in a second. So for just drawing on that last example, the Virica example, this is where we have a panel of 4 to 5 curated molecules for use in what we're calling proprietary and commercial platforms. So these might be platforms where CDMOs or particular therapeutic developers have gone to the original pool of HEKs, pulled out some sort of proprietary cell line or unique cell line. We've got these 4 to 5 molecules, which we have seen be beneficial across a broad range of AAV production to look at in that given system. And we have very nice defined dosing ranges in terms of cytotox. We validated these already for suspension and adherent systems. And we consistently see in between that 2 and 4x yield at the bioreactor scale. And important to note that we do tend to be serotype agnostic, i.e., once you have a formulation developed for your cell line or whatever cell line platform you are using, that switching serotypes does not materially change that formulation. You can still expect that 2 to 4-fold increase at the bioreactor scale, regardless of serotype. But specifically, too, we have also validated or pre-optimized these molecules for some of the key manufacturing platforms. And this is an example of the Thermo Fisher VPC 2.0 cell line, again, with Fectavir and other PEI-based reagents. This is where we have a curated set of 2 molecules, 007 and 008, which consistently provide in between that, again, 2 to 4-fold yield at the bioreactor scale when used in the viral production media that's generally recommended for these cells, again, with PEI-based reagents. Additionally, anyone out there using the NRC HEK suspension cell line, this is actually one of the examples that we took you through where we saw that 4-fold or almost 4-fold increase based on a single molecule. So this would be a single molecule that we can hand off to clients in this particular platform. Again, validated for use in the production using NRC's HEK suspension platform with PEI-based reagents. In between that 2 and 4x yield across a couple of different serotypes, specifically when used in the balanced CD media. So again, drawing on our knowledge and all the different kind of platforms we've looked at, we can really kind of tailor a curated set of molecules that we think will be beneficial within any given AAV production platform, whether it be suspension or adherent. So really key takeaways for VSEs and the use for AAV production. Typically we see between that 2 and 4x boost when they do work on these HEK-based systems. You can look forward to actually VSE-007 being available as an off-the-shelf molecule, specifically for VPC 2.0s with Vectavir and other PEI reagents. That will be launching later this year. It's important to note, again, too, here, the most salient feature when developing VSE formulations is the actual cell line. The cell line itself dictates which of the chemical classes are most active. Once a VSE formulation is developed for that cell line in a media with a transfection reagent, basically you can start switching out the serotype, you can start switching out the transgene, knowing that that formulation will by and large give you a meaningful benefit. And again, here, Virica's high throughput screens really allows us to pull out those molecules, those panels, as I kind of allude to them, to really help optimally identify VSE candidates for AAV production. Okay, so I think we're going to pause or stop on the AAV and now speak a little more towards cell therapeutic applications. So again, here we're going to switch gears. We're going to talk a little bit about lentiviral production, but we're also going to talk about trying to bring efficiencies into the transduction step. And so when we think about the developing cell therapy applications, there's really two parts where we think the VSEs can be beneficial. And then we're kind of looking to bookend that manufacturing process, or at least the parts that involve the introduction of nucleic acid or viral vectors. And the first angle is obviously through viral production. So kind of as alluded to with the AAV, the VSE technology can be very useful in these reverse genetics type systems. So here we'll talk a little bit about how VSEs, and again, it's a different panel of molecules that we've curated here specifically for lentivirus. So I'll walk you through that a little bit. That can really be beneficial in the production of functional lentiviral particles. Now, I do want to point out here, this graphic sometimes bothers me because those are clearly not lentiviral particles. Those are AAV particles. However, you can see the concept will basically translate to lentivirus. The second area where the VSEs can be really beneficial is in that transduction step. So if we take a moment and think back to our central hypothesis that there are these biological barriers to these steps in the generation or the manufacturing of a cell therapeutic, you can be sure that those primary cells, whether they be the activated T cells, NK cells, stem cells, these cells are going to have fully intact suites of innate antiviral responses. And so that same hypothesis holds true here, that the use of our molecules prior to or at time of transduction or introduction of that nucleic acid can transiently attenuate those potential biological bottlenecks and increase transduction efficiency. So we're going to take you through some of our new data that are indicating or demonstrating the utility of the molecules in both of these steps. So very similar to what we did with the AAV is we developed a high throughput lentiviral production screen. And again, screened over 16,000 molecules looking for hits that gave us a statistically meaningful 1.5 and above increase in the functional titer of these lentiviral molecules. So again, on the right-hand side, I'm just showing you kind of the output of the primary screen. We take the hits from that. We do hit validation. We always lose a few during the validation process, but basically take the winners out of that validation process and really enter them into our product development pathway, where we look at their utility across different modalities, shall we say, or different platforms for lentivirus production. So here again, we've curated a set of about 4 molecules that are useful in both adherent production in HEK293 T cells, as well as a set that is good for suspension production in HEK293 lineages. So what I can show you here is on the left-hand side, this is obviously a panel of 4 molecules labeled VSE E through J, which in the adherent, and this was tested across 2 different lineages of 293T adherents, where we see, again, in that 2 to 4x range when we talk about functional titer. So this is your standard HT1080 assay as our readout, reading the functional titer out of these production systems. And then on the right-hand side, this is in suspension VPC 1.0. So again, now we're in an HEK lineage in a suspension format, where you can see VSE-E, which was our top candidate in the adherent system, also gives us in that 2 to 4-fold increase in functional titer out of the suspension systems. So regardless of which of these two platforms you're using for the production of your lentivirus, we will have a curated set of anywhere between 2 and 4 molecules, which we think could probably provide meaningful benefits in terms of the increase in functional titer. So again, here in that suspension system, seeing that nice 4x boost in titer out of the VPC 1.0s. Now, again, too, recognizing that the lentivirus field in particular, and this is true of AAV as well, their lentivirus tends to be a little more mature in this space, is that some people are shifting to the use of packaging or fully inducible producer cell lines. And so one of our questions, again, here is the format's a little bit different, the platform's different. Could the VSEs be useful in this particular context as well? So in this study, this is where we partnered with an organization who had a fully inducible suspension HEK lentivirus platform. So this is one where all the structural components, as well as the gene of interest and the genomic component, are integrated within the cell. And then there is a small molecule induction. They use a small molecule to induce the production of this lentivirus. When we looked in these producer systems initially, we actually added our VSE molecules at the time of induction. And what we saw there is absolutely no benefit. Every titer came back pretty even Steven with the baseline. And that prompted us to kind of go back and think about the kinetics of the innate antiviral response. So if you'll recall, in that very nice paper done by Eric Barton and the team at Pfizer, there was that incoming nucleic acid that set off the interferon response, and then you got that culminating in the innate antiviral response. We had to think about that in the context of inducible system. There is no longer that incoming nucleic acid from transfection. It's really all about the byproducts of assembly and replication. So here, this particular platform, they were using an HDAC inhibitor around the 18-hour post-induction time point. And so what we did is added the VSE molecules at the same time as that HDAC inhibitor. Again, just a little bit of a kinetic difference in terms of when we use those molecules. And that's what we're showing on the right-hand side here, where of the 4 molecules we tested, 3 of them gave that very nice statistically significant increase in functional titer out of this inducible system. So again, here, curated set of molecules for inducible systems for lentivirus production. Now, finally, I will just talk a little bit about the use of VSE technology or small molecules technology for lentivirus transduction. So here we did a minimal screen for LV transduction enhancers in activated T cells. Again, looking for things that are in that upper right quadrant, which give us a statistically meaningful benefit to transduction efficiency. And what we did is, out of that particular screen, we hit on 2 compounds, compound A and B, which provided about a 2x increase in the transduction efficiency of lentivirus in activated T cells without providing any meaningful impact on viability, which is what we're showing on the right-hand side there. So what does this mean for transduction efficiency? I would like to actually, sorry, I should probably give you a few details here. This is a VSV pseudotyped lentivirus on activated T cells. And we were able to show this 2-fold increase, and in between 2 and 3-fold increase across 3 to 5 independent donors. So very consistent benefit to these 2 compounds without materially affecting viability. So really, again, you can think of these as tools in the toolbox to allow you to do one of 2 things. And so I'm going to just walk you through this slide a little bit. One is you can just increase the transduction efficiency of your process to hit more of those T cells to try to get to your therapeutic dose quicker, or you could use the VSEs to dose spare some of that precious lentivirus. And that's actually what we demonstrated in this particular study. So I'll walk you through this step at a time. On the right-hand side, you'll notice the MOI is 6. And on the left-hand side of this graph, the MOI is 2. What I'll draw your attention to is the dark blue. This is the baseline transduction efficiency. So as you can see, for the MOI of 6, baseline transduction efficiency of about 40%. And then at an MOI of 2, we see that baseline transduction efficiency drop to about 15%, as would be anticipated with a lower MOI. You can see at the high MOI, the use of VSE A or B, shown in the light blue or the gold, does provide that statistically meaningful benefit. We fell a little short of the twofold here with this high MOI, but a very nice benefit of those molecules in that higher baseline transduction efficiency. But what gets interesting is you'll note the level of baseline transduction efficiency when the VSEs are present in the lower MOI group. So the MOI2 group, again, baseline dropped to about 15%, but with the VSEs, we were able to hit just above that 40% threshold. So what this actually means is with the VSEs at an MOI of 2, we are getting the same baseline transduction as we do at the MOI of 6, showing a 3x improvement, which allows you to dose spare that lentivirus. So depending, again, on where your bottlenecks are, you can either use it to increase your transduction efficiency or use the VSE technology to dose spare your lenti, or maybe something in the middle, whatever makes the most sense to your process. And again, here, this is where we really use or view the VSEs as just tools in the toolbox that allows you to really dial in your platform process in the way that's most meaningful to you in terms of the COGS or the efficiency of that process. So here, again, key takeaways for cell therapy. We can boost lentiviral production, again, in titers up to 4x, as we demonstrated in the VPC 2.0, or sorry, that was the VPC 1.0 cell line. We've seen up to 3x and greater transduction efficiencies without impacting viability when we're talking about the cell side of things, and allowing you to reduce the MOI needed to achieve meaningful transduction within those platforms. So again, Virica can leverage those high throughput screens and our library of small molecules to rapidly identify candidate molecules to help in the cell therapy type platforms. All right, so I'm going to pause there. We're going to stop talking about VSEs a little bit, and we're going to move on to something that we've been leaning into a little more here at Virica, and that is leveraging design of experiments, DoE, but in particular, marrying that together with our high throughput virology services. So really, this is where we've developed a lot of core expertise on the VSE library, developing high throughput virology methodologies, and using these DoE strategies to accelerate success. And we leverage these together, and we can really use them as tools to rapidly optimize upstream production parameters, as well as develop application-specific VSEs. So I'm going to contextualize all that over the next 5 minutes or so, but really, what is DoE? So DoE is a systematic approach. It's really a statistical approach that allows you to assess multiple factors affecting a system at any given time. And this is really, again, based on statistics. So if we take traditional approaches such as OFAT, one factor at a time, you can kind of think of your design space as a 3D cube. You can kind of look at all the axes, so X, Y, Z, one at a time, and optimize a system, by looking at one thing and then stacking on top of that and then stacking on top of that. It's a slow process, doesn't allow you to look at everything, but has been the traditional approach, especially within biology. When you use DoE, you actually open up that entire three-dimensional design space by using carefully designed and statistical-based approaches. So really, how is this beneficial to viral vector production? Well, when you talk about these reverse genetic systems, AAV production, lenti production, there are lots of different inputs to look at, not just VSEs, right? There's transfection reagents, there's medias, there's different cell lines, cell types, there's ratios of your transfection reagent to total DNA to cell, the ratios of the plasmids together. It's actually an overwhelming amount of combinations. Just for fun, we did do back of the envelope calculation, and there's with the commercially available reagents, there's over 19,000 different combinations to look at. And that's just completely unreasonable using those one factor at a time approaches. And quite frankly, DoE will struggle to look at 19,000 as well. However, it does unlock more of that design space. And so here at Virica, we've been leveraging this using our high throughput virology. I still do things in triplicate. So using one iteration of our DoE, we can look at 360 unique conditions. This is over 1,000 data points because, as I mentioned, we still do things in triplicate here using a functional titer readout. But what this allows us to do is not just look or design VSE formulations optimized for any given platform. We can look at some of those other inputs to truly optimize an upstream platform. So this is an example of a DoE that we did using our VSEs, but also looking at 6 additional input factors, including plasmid ratios, plasmid sets, transfection reagents, total DNA, cell densities, and DNA ratios. And really, this allows us, irrespective of the VSEs, to really rapidly develop upstream processes, which we've now demonstrated at the bioreactor scale for AAV, lentiviral, or quite frankly, replicating viral vectors as well. So kind of a unique marrying of our high throughput methodologies with our DoE methodologies, again, to really unlock the full potential of an upstream process and demonstrate validation. So we can take it as far as the 250 mil bioreactor, but we've seen really nice translation from our 96 well DoE to our 250 mil bioreactors, and now with some partners beyond that scale. Finally, I do just want to talk about a little bit of the benefits of these molecules to the manufacturing process. And sometimes this is something we're not always thinking about when we're doing that upstream process. But by maximizing your yield or really developing a robust process at the beginning, it can have absolutely large impacts. And whether that's increasing the accessibility of these life-saving strategies that a lot of the therapeutic developers are going after. And again, that's kind of the end goal here, is like bring some options to patient populations that don't have any therapeutic or very limited therapeutic options. But we have to keep this in mind that it's got to be at an accessible cost as well. So here, through using DoE strategies and VSE, really optimizing that upstream to get that global impact, that financial impact, it's something that you don't always think about, but that environmental impact as well. When you see the amount of plastic waste or even water usage that goes into bioreactors, by reducing those footprints, we can impact all those different scenarios. And so we do some cost modeling, again, showing the benefits or trying to articulate the benefits of truly optimized upstream processes to the patients and to organizations as a whole. So this is, and again, we use Biosolve through Biopharm Services to do this cost modeling with our partners. This is an example where we were able to achieve a 4x increase for this particular treatment. And what we show here is that 4x increase is actually able to decrease the dose or the treatment cost of this particular therapeutic by about 75%. So for example, here, what that meant is per batch or per production scenario, per bioreactor run, they were normally getting about 9 doses, 9 patients being able to be treated. By that 4x yield, again, accounting for some downstream loss, as well as some of the material needed for analytics, we're able to increase this to 34 patients per batch. And that's an overall 75% reduction in the per dose cost. So this really allows a lot of flexibility in terms of the pricing strategies for these, gets broader market penetration, and again, importantly, that increased accessibility to patients, because you're actually also not only reducing the cost, you're getting to the doses you need quicker and faster as well. And so another way to view that is on the per batch, but also on the long-term cumulative savings for a commercial product. So we always like to plan for success, right? So obviously, if you take that 4x or taking the time to optimize your upstream yield at the very beginning, what does that mean for the lifetime of the product? And so again, that same scenario that we modeled over 10 years based on market penetration and all that other good stuff can lead into the hundreds of millions of dollars of savings over the commercial lifetime of product. In this particular case, it was 578 million USD in savings over a 10-year span. Again, even accounting for the cost of implementing the VSEs, which is quite negligible compared to those potential savings. So this allows you to free up a lot of your resources for R&D pipeline expansion, again, enhanced ability to innovate and maintain competitive market positioning, and really reaching that commercial scalability and viability faster. Okay, so I left you guys with a lot to think about and a lot to absorb. Ways to work with Virica. Well, obviously we provide products and services. So products mainly centered around that VSE technology, off-the-shelf VSEs for VPC 2.0 coming soon. But again, AAV production, LV production and transduction, vaccine production, replicating viruses. We can help in all of these different modalities through two ways. Either what we're calling client-driven evaluations, where we MTA, we will send the molecules to you. You get a dedicated support team to help design your experiments and walk you through the process. Or again, we are happy to onboard your platform into our labs, which are located actually up in Ottawa and Canada. And we can leverage all of our workflows there to rapidly develop upstream and VSE processes for any given platform. Additionally, again, DoE scale up. We can do process modeling, cost modeling. We have a lot of analytical services, including residual testing, viral quantification, and transduction assays. So if any of this resonates with you or you have more questions, please feel free to reach out to us anytime. And at this point, I will pass back to Eden and we can do some question and answers and discussion. So I'm not sure if anyone else is getting this, but I'm just getting a lot of feedback right now. And sorry, we weren't able to hear you there, Eden.
Oh, right. Oh, sorry. Okay, let's try again. Okay, let's see if that is any better. That was a brilliant presentation. We're just going to give people a moment to enter their questions. But in the meantime, I would like to thank Virica. Okay, it seems I still can't be heard.
You know what, Eden? Maybe just considering what's happening, I'll just read the questions if that's okay with you, and then kind of address them one by one. Okay, so I'm just going to walk through some of the questions we've got through the chat here. Can you please comment on the impact of VSEs on AAV quality profile, specifically talking about product-derived impurities, including full empty, packaging of undesired plasmid, host cell DNA, et cetera, et cetera? Yeah, really great question. And I think this is something, obviously, that is becoming almost, I don't want to say a more important conversation than yield, but certainly top of mind. Because you do want a good yielding system in terms of overall bulk, but we are learning that product quality is probably a more important step, at least when it comes to the efficacy and safety. So yes, when we think about those effector molecules that are part of the innate antiviral response, their whole job is to actually try to destroy those nascent virions. And the macromolecules that go into making nascent virions. So they're, nicking up nucleic acid, trying to destroy nucleic acid. They're tagging proteins in certain ways that are either causing them to be degraded, partially degraded, or try to render them inactive. So what we've seen, and it's really tough to measure. It's really tough to measure sometimes some of these responses, but what we've seen in certain contexts, I think I provide the one example where we do see an increase in that full empty ratio, and we've seen that across a couple different platforms. Not every platform, to be fair, where we get about that 2x increase in full empty. We've never seen a negative impact of the VSEs on full empty, always a positive or neutral impact. But we've also seen other areas of the quality go up. This includes decreased host cell DNA contamination. We think this has to do, again, with perhaps apoptotic pathways not being activated in the presence of the VSEs. So you don't get that nicking up of nucleic acid, the fragmentation, which can be those process impurities that come through. So we've certainly seen full empty go up in certain contexts. We've seen host cell DNA come down in certain contexts. I really want to get into the packaging, Our next step with some of our VSEs is to do some NGS on a VSE versus non-VSE treated population to really look at the impacts on the genome. But certainly that was a really long-winded way of saying we have seen a positive impact on those quality aspects. Just a little more work to be done, because obviously when you talk about quality, there's different metrics, and being able to explain how the VSEs are impacting those metrics is something we're definitely digging into. Another great question, I think, is potency. We have seen where it appears we are generating more potent vector under certain scenarios, but again, proving that out is always a tricky scenario, because potency is a tough one to measure with certain serotypes. But yes, certainly. All right, another question. What transfection reagent did you use with your AAV VSE screen? Yeah, great question. So by and large, we do find most people lean towards PEI-based approaches. And so when we did our screen, we used PEI-based reagents to do that screen and the validation. So these tend to be one of PEI Max, PEI Pro, or Effectivir, depending on how we're approaching the screen and or the validation. But certainly really sticking with the PEI-based reagents. Now, that being said, we have seen the VSEs be functional in non-PEI based reagent platforms as well. It's just you might want to screen a couple more. So like, for example, I probably wouldn't send 2 molecules for that. I'd send a panel of 4 to 5 molecules, just because that is a significant process change. On slide 13, what was the AAV productivity of the control in VG per mil? Oh, geez, I'd have to remember what slide 13 is. Hang on 1 second. Let me just scroll back up if I can. Slide, okay. You know what? I'm going to treat this one in a more general context. So most of those examples that we worked through for AAV, the baseline production, again, depends on the serotype, right? The serotypes have different baseline productions. But for all of the case studies I walked us through, the baseline productions have been mid to high E10s into probably the highest baseline production scenario in those therapeutic applications that we talked about, into like the 3E11. So probably, you know, mainly in between that 6E10 to 3E11 range for most of the examples that we provided. The higher the productivity, the more challenging it is to drive up the yield. Yeah, really good point. Yes, and this is true too. That the higher the baseline, it's absolutely true. It's the more challenging to drive up the yield. However, that being said, in some of our higher, and again, everyone's high producing baseline is different, so I will contextualize this. Even in systems where we're hitting into the E11s per mil, just want to be clear on that, we are still seeing, you know, in that 2 to 3 and sometimes 4x improvement in the yield. But I think your point's well taken, that the lower the baseline, the more room for improvement, certainly, especially when we're talking about DOE type strategies and the VSEs. All right. Do we still have time? I'm assuming we still have time for questions. So I'm just going to keep going until they basically shut us off. Okay, perfect. We still do have time. What are some examples of how DOE has helped identify unexpected but beneficial interactions? Okay, another really good question, because a lot of the folks who talk about DOE strategies talk about uncovering what they call hidden interactions. And really what they mean by that is essentially when you're going one factor at a time, you can't necessarily detect synergies between 2 different inputs. However, when you have that 3D design space, and when we talk about DOE, this is where you can hit on those unexpected or so-called hidden interactions. So I do actually have a real-world scenario case for this. Again, you'll just have to bear with me. Before we were using DOE approaches, we would generally look at a panel of VSEs as single molecules first. And so let's say if we started with a panel of 6, we would look at 6 molecules. We would pull out the ones that we thought were giving us a 2x or better increase in production. And then we would triage away molecules that weren't providing that 2x. and we would start bringing those ones that were functional together in pairwise combinations in a second step. So that was prior to using DoE. When we switched to using DoE, this allows us to look at single molecules for sure, but all the different pairwise combinations in a single experiment. So we did actually have an experiment in a replicating virus where two molecules on their own were only giving about a 1.5x increase in viral vector yield. We would have normally triaged these molecules away if we weren't using a DoE type of approach because we had better hits. However, when those two molecules were brought together in, our DoE, we actually got a 7x increase in viral vector yield. So that was actually a synergy between two of our molecules. Now, we don't always pick up synergies. This is just one of those happy situations where we got synergy between those two molecules, and that's something we would have missed in our traditional or our one-factor approaches that we were able to reveal for using those DoEs. So I would say that would be the standout one in my mind. Okay, staying on DoEs, we've got, okay, at what stage is DoE implementation optimal? Okay, I'm gonna say it's never too late to do DoE. However, since that question here is optimal, the answer would be as soon as possible, really while you're trying to do your baseline. And I only say this because, you know, when developing a platform manufacturing, eventually you're going to get to a point where that platform is going to get locked. You know, hopefully you have a clinical asset that is marching through those stages and you're going to have to lock in. Now, are post-BLA changes, you know, accessible? Yes, absolutely they are. In fact, you know, we've had conversations with folks who, you know, maybe want to use the VSEs post-BLA. But there are barriers, quite frankly, there. You know, there are a few boxes you're going to need to tick, especially regulatory-wise, which we're well prepared to support. But when you're talking about DoE, where you might be changing more than just adding, you know, an additive into the system, but fundamentally changing how you're approaching the production, you know, post BLA might not be the spot. So I would say, you know, you're going to want to implement DoE sooner rather than later to really nail down a very nice upstream platform. And again, you can think of that as a platform that you can build on for all of your clinical assets, right? Understanding your platform using DoE, using, you know, an AAV2 serotype is still really going to be informative when you switch to AAV8 for your next clinical asset or some sort of proprietary capsid, perhaps. Having that really good understanding of the inputs and what drives your system using DoE can be really beneficial across your whole pipeline. Okay, I'm going to move on here. Let's see what else do we have here in the chat. Okay. How quickly can VSEs be adopted? Oh, yeah. Okay, so this is all going to depend on what you're working with. Again, if you're in, for example, VPC 2.0s with Vectavir, we're going to hand you two molecules, very defined dosing ranges. You're going to be able to do a study quite quickly and realize the benefits there. If you're in a more of a proprietary cell line, there's a little bit more work, but it's still not accessible, right? We'll probably send you anywhere between 2 and 5 molecules. Again, nice defined dosing ranges. But really what gets interesting there is if you can put in the time and you get more than one hit, you can bring those together in the pairwise combinations, as we demonstrated with Virica, to really get those additive benefits. But once you get that formulation, you know, our VSEs are available in both R&D and GMP grade. So if you're happy with that upstream process and you want to walk it down the hall to your GMP and your manufacturing folks, you know, you can straight walk that R&D grade assessment, and we're ready there for the GMP process. So again, if you're in a really well-defined system that we have a deep understanding of, it can be adopted very quickly, but we still support you if you're in a proprietary line. And I think that's probably the cue for me to stop talking. Appreciate everyone today. And again, please reach out if you have any questions.
That's all the time we have for questions, but thank you for a great session. Thank you, JD. If anyone submitted any questions that weren't addressed, it actually looks like we addressed all those questions, but keep in mind the speaker will reach out to you directly if there were any questions that weren't answered. This session was recorded. You'll receive a notification within 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to download the white paper in the Resource Center to the right of your screen. Please also take a moment to complete the feedback form so that we can improve on your webcast experience. On behalf of Informa Connect, I'm Bradley. Thank you so much. Have a wonderful week. And thank you very much to John David.
Thank you.